Happy morning. Today's topic for discussion is on genetics. Genetics is a study of heredity and variation we know. Today we are going to deal about the microbial genetics, which is a branch of genetics concerned with the transmission of hereditary characters among the microorganisms. So this microbial genetics had a wider application in the fields of molecular and cell biology and in the field of medicine, agriculture, food and pharmaceutical industries. So every one of us know the central dogma of life is DNA, which is transcripted to RNA and translated into polypeptide. So it was discovered by Watson and Crick. So the basic structure of DNA is proposed by Watson and Crick. We know it is a double helix model and it is composed of two chains of polypeptides. Each chain has a backbone of deoxyribose sugar and phosphate residues arranged alternatively. And the four nitrogenous bases in the DNA are two pyrimidines, including adenine and guanine, and two pyrimidines, thymine and cytosine. So this is a double helix model with the alternating bases. When coming to the structure of RNA, it is structurally similar to DNA except for two major differences. One is it is made up of ribose sugar, and in the place of thymine, uracil is there. And when coming to the types of RNA, there are different types, including messenger RNA, transfer RNA, and ribosomal RNA. So we all know DNA is a complex structure which makes life. And the unit of hereditary is gene, which is a segment of DNA specifying for a particular polypeptide. And we need to know about certain terminologies. So one among the important one in genetics is codon. So codon is nothing but a unit consists of sequence of three bases. So codon is a triplet. A code, single code can make a single amino acids. There are nearly 64 codons, more than one code present for making similar sequence of amino acids. We all know there are few amino acids, but we have 64 codons. So more than a code can present for a single amino acids. So these are the examples, like AGA will make arginine, and the same amino acids will be having different codes like AGC, CGU, CGG, also code for a similar amino acid arginine only. And there are very few codons which will never code for any amino acids. So three of them are UAA, UAG, and UGA. These codons are known as nonsense codons or stop codons. So whenever you come across these codons, the synthesis of the polypeptide stops there and there will be a termination of the polypeptide synthesis. Hence, it is known as stop codons or nonsense codons. And coming to the other terminologies, introns are nothing but the non-coding sequences on a gene, and exons are the coding sequences on a gene translated into gene products. When coming into your bacterial cell, bacterial genome, it is nothing but a collection of all the genes present on the bacterial chromosomes or its extra chromosomal genetic elements. As that of an animal cell, bacteria will never have a central nucleus, nucleolus, nuclear material, everything. It just as a chromosomal material in the center, along with the extra chromosomal genetic element, which is known as plasmids. So the bacterial central chromosomes, generally it is a double-stranded molecule of DNA arranged in circular form. And in very few cases, it will be arranged in linear form. For example, Streptomyces, Agrobacteria, or Borrelia. Apart from this central chromosomal material, we do have some extra chromosomal genetic elements, which are not essentially needed for the survival of the bacteria, but it confirms to the properties like drug resistance, toxin production, etc. So these are nothing but the plasmids. They are the circular DNA molecules present in the cytoplasm of the bacteria, and they are capable of autonomous replication. So it can transfer genes from one cell to other, and thus we are using this plasmids as a vectors in genetic engineering. So when you see this picture, the red ones are the central chromosomes, bacterial chromosomes, and the blue circular extra chromosomal materials are the plasmids. So here we can find the integrated form of plasmid with the chromosomal DNA. So that is known as episome. So here comes the different types of plasmids. Plasmids are differentiated into many varieties based on the properties. So based on the ability to transfer, it can be a conjugative plasmid or non-conjugative plasmid. Based on the property for which they code or for which they produce, they can be of R plasmids, which code for drug resistance, F plasmid for fertility factor, Col plasmid, which codes for carcinogenic factor, and many plasmids will code for the virulence factors of the bacteria, and many will code for degradative enzymes. And based on the incompatibility typing, we can call it as incompatible plasmids or compatible plasmids. So here is an example for the structure of R factor, that is resistance factor. So when you see here, there are two components. One is RTF and R determinant. So this R determinant will code for the drug resistance, and this RTF is nothing but the resistance transfer factor. Not only the RTF is present in this molecule, this whole drug resistant determinant can be transferred. Without the help of RTF, it cannot be transferred, but it is complete with the presence of RTF. And there are some other genetic materials in the bacterial cell. They are known as transposons. These transposons are nothing but the insertion sequence present in the plasmids or the chromosomal material, and they have a special property of jumping here and there. It can detach from one place and get attached to another place on its own. So that is why it is known as jumping genes. And this movement of the insertion sequence within the bacteria or among the bacteria is known as transposition. So this is a structure of the transposons. It will be similar to a lollipop structure. This inverted sequence is readily detached from one part of the chromosome and it is readily attached to the other part. That is why it is called as jumping genes. So while coming to the functions, we know that RNA transfers the genetic information from one generation to another. RNA directs ribosomal synthesis of polypeptides by translation. And as we saw, plasmids, they are coding for the fertility, resistance to antibiotic, radiation and phages, production of various exotoxins, endotoxins, bacteriocins and enzymes, and they will be causing for tumorigenesis in plants. And coming to the transposons, it contains many antibiotic resistant genes. They are the major factor in development of plasmid conferring multiple drug resistance. And where these are applied. So as we discussed earlier, plasmids are generally used as the vectors in genetic engineering. They are very much helpful in the production of a lot of medicinal products like insulin. And this plasmid liposome complex, they are generally used for gene therapy. And transposons also, they are used in genetic engineering. Multi-drug resistant plasmids produced by transposons, accumulation of single plasmid is the major cause for drug resistance. And they are the cause of spreading antibiotic resistance. And coming to an important topic, bacterial variation. So variation, as we all know, some differences in the properties, right? So that may be of phenotypic variation or genotypic variation. Phenotypic is nothing but some variation in the physical appearance. Genotypic variation is some differences in the gene level. And there are few differences between these two variations. So as we know, it is a physical expression of the genotype in the given environment. Phenotype is a physical expression of the genotype in the given environment, and genotype is a genetic constitution of a cell that is transmitted to its progeny. The difference between these two are the phenotypic variations are generally influenced by the environment. They are not generally temporary and not heritable. When compared to the genotypic variation, they are not at all influenced by the environment. Generally, they are stable and heritable. So these characteristics expressed by a bacterial cell at a particular time, temporary not heritable, and influenced by any of the environmental factors. So the bacteria exhibit different phenotypic of appearance, and they are generally reversible. When you change the environment, the phenotypic variation also changes. For example, Salmonella typhi will synthesize flagella for its motility, right? When it is grown in a phenol red agar, this flagella synthesis is inhibited. So it is inhibited by the environment, right? So this is an example for phenotypic variation. Another example is beta-galactosidase enzyme is produced by E. coli only in the galactose medium. So we have certain constitutive enzymes which are not influenced by the substrate and certain inducible enzymes which are generally influenced by the substrate. So this beta-galactosidase is one example for this inducible enzymes. So it is produced when only the medium is containing galactose. So these are examples for phenotypic variations. And coming to the genotypic, that is genetic variation, we know that they are stable, heritable, and not at all influenced by environmental factors. So this genetic variation can occur due to mutations or gene transfer. So mutations are nothing but random, undirected, heritable variation caused by alteration in the nucleotide sequence at some point of DNA of the cell. So these mutations can be grouped into two separate groups. They may be of spontaneous mutation, or sometimes it is induced by the external factors. Generally, these mutations can be harmful, lethal, and many times it is helpful and silent also. So coming to the types of mutations, we do have plenty of mutations. For example, point mutation. Here, only a single nucleotide is replaced by another. Example is addition of a single nucleotide, deletion of a single nucleotide, translocation or inversion. And even base pair substitution will occur. For example, transition and translation. Apart from that, silent mutations are there. So here the change at DNA level that does not result in any change in the amino acid in the encoded protein, as more than one codon may encode a single amino acids. So there will be no change of this type of mutation. And the next type is frame shift mutations. So during replication, one or few adjacent base pairs have been inserted or deleted. So the frames are shifted. So these are the examples for addition. Here you can see addition of few nucleotides like DE is added again here. Deletion of certain nucleotides, translocation occurs here, and inversion can be seen here. So as we discussed frame shift mutation, this shifts the reading frame and coded message is read correctly up to that point, but the subsequent codons specify a wrong amino acid. And the other type is a missense mutation. So the mutant base pair changes the coding sequence, resulting in the production of a different kind of amino acids. And as we saw in the earlier slides, nonsense codons, which codes for a termination of protein synthesis, they will cause nonsense mutations. Example, UAE, UAG, and UGE. And there are multi-site mutations which will cause macro lesions involving large number of base pairs. And there are some helix distortion mutation also, which is induced by the UV radiation and dimerization of thymine. And certain mutations are suppressive in nature. They restore the function of the gene inactivated by the previous mutations. And certain mutations involves the vital functions so that the organisms are killed. So those mutations are known as lethal mutation. And conditional lethal mutation affects the secondary or tertiary structure and respond differently. For example, temperature sensitive mutant, which is able to live at permissive temperature, but not at restrictive temperature. So it can live at 35 degrees centigrade, but not at restrictive temperature. So here are some examples of mutagens. So physical agents like ultraviolet radiation, ionizing radiation, even visible light and heat can cause mutation. On prolonged exposure, mutations can occur. And chemical agents like alkylating agents, acridine dyes, bromouracil, and aminopurines are also can cause mutations. And we do have certain tests for mutations like fluctuation test and replica plating method. So under genotypic variation, we were reading about the mutations and gene transfer. So we are done with the mutations. We'll move on to gene transfer. Gene transfer is nothing but transfer of genetic material from one bacteria to the other bacteria by various means. So 4 different types of gene transfers are there. One is transformation, transduction, lysogenic conversion, and conjugation. Transformation is nothing but transfer of free DNA from a dead bacteria to the other one. So just a transfer of free DNA from a dead bacteria to the living bacteria is known as transformation. So the transformed free DNA will get incorporated into the living cell and they will start producing their own property in the living cell. So this transformation is proved by the scientist known as Griffith. He did an experiment on pneumococci. What he did, he took a live capsulated bacteria, that is pneumococci, into a mouse. So this capsule is a virulence factor for pneumococci. So it is a major cause of the disease and hence the mouse is killed of infection. So when live capsulated bacteria is injected into the mouse is killed. In the second step, what he did, he introduced a live bacteria, but it is not capsulated. That means the virulence property is removed. So in this case, the mouse is not getting any infection and it is still alive. In the third step, what he did, he killed the bacteria with capsules. Though the virulence factors are there, but the bacteria is not alive for replication. So again, the mouse is free of infection. So to reduce the virulence, what we have to do? Either we have to remove the virulence factor or we should kill the bacteria, right? So what Griffith did, he combined these 2 steps, second step and third step. That is, he mixed a living bacteria without capsule and a dead bacteria with capsule. So what happens in this dead cell? The gene coding for capsular production is transferred to the living cells, which is not having any capsules. So this property is transferred from the dead cell and this living bacteria will start producing capsules. So what happened? On further replication, the bacteria is able to synthesize capsules and this encodes for the virulence factor and hence the mouse is developing the disease and it dies of infection. So this is how Griffith proves the transformation. So the second method of gene transfer is transduction. Here the DNA is transferred from one bacteria to another by a virus known as bacteriophage. There are 2 different types of transduction. One is generalized, any segment can be transducted, and restricted, only a particular segment can be moved. So this bacteriophages, they do have some different life cycle in different bacteria. Okay, so they have a lytic cycle and lysogenic cycle. So this transduction involves the lytic cycle. When a virus, which require a living cell for its replication, generally enters the bacteria, injects its nucleic acid into the bacteria. When the nucleic acid is introduced into the cell, what does it do? It stops all the machinery of the bacterial cell. It stops all the functions and metabolism of the bacterial cell. It starts producing its own products inside it. Okay, so only the nucleic acid and protein part of the viruses are done in the bacterial cell using all its resources. So it starts producing plenty of nucleic acids and plenty of proteins. And finally, what happens? They will undergo assembly. So everything is packed. But while packing, what they will do, they will take a fragment of this bacterial genome also with them. So this bacterial genome is also packed in the new virions. And when they are leaving the cell, they will kill the cell and leave. But while leaving, they are taking the bacterial genome also with them. When they are going to infect a new bacteria, the property of the mother cell is transferred to the new cell. So this is how a bacteriophage, a virus, is transferring a segment of gene from one bacteria to the other. So this type of cycle is known as lytic cycles. This is known as transduction. So it involves chromosomal DNA, episomes, and plasmid genes. Any group of bacteria for which bacteriophages exist can be subjected to transduction. So as we discussed, restricted transduction adds to the virulence of bacteria. And the method is used in genetic engineering in correcting certain inborn errors also. And this is an excellent tool for genetic mapping. And we also saw that it has a different life cycle, that is lysogenic conversion. So what happens here? The same thing, the bacteriophage, the virus enters the bacterial cell, injects its nucleic acid inside, and when instead of producing its own nucleic acid and all the other machineries, what does it do? It gets incorporated into the bacterial genome by itself. So it is becoming a prophage, that is known as prophage. It is getting incorporated into the mother cell's DNA and forming a prophage. So when this mother cell is replicating, the property of the viral DNA is here only. So the whole cycle is repeated. So this bacteria is acquiring the property of the bacteriophages. Sometimes these phages may acquire this DNA from other bacteria also. So this is known as lysogenic conversion. So when you see here, this is a diphtheria bacillus, and when it is normally diphtheria bacillus will not produce any toxins. When it is infected by a beta phage, a virus, then only it is able to produce a toxin and cause disease. And the last method is conjugation. Conjugation is nothing but the transfer of genetic material from one bacterium to another bacterium by mating or contact. A conjugation tube is formed in between two bacterial cells, and whatever genetic material that are to be transferred will be easily transferred to this other cell by the conjugation tube. So all the types of plasmids can be transferred by conjugation, especially F factor, col factor, and RTF. So we know that col factor is coding for colsin, piocin, and diphtherosin. And we discussed about this resistant transfer factor also, which has a R determinant and RTF. Without this RTF, the whole component cannot be transferred. And we also know about this transposons. They are nothing but the jumping genes. When the insertion sequence are moving here and there, it is known as transposition. This is also an example for gene transfer. So the application of this gene transfer is, they are used in the recombinant DNA technology, gene mapping, transfer of drug resistance, and transfer of certain virulence factors. Coming to an important topic of drug resistance. So what are the different mechanisms of drug resistance? A bacteria can acquire drug resistance by genetic mechanisms, which may be by mutation or by gene transfer, or through some biochemical mechanisms, reduced permeability of the drug, developing an alternate metabolic pathways, and producing an enzyme inactivating the drugs. So these are the various methods by which an organism is acquiring drug resistance. So we all know the genetic mechanism behind drug resistance is mutation and the gene transfer. Gene transfer includes transformation, transduction, and conjugation. But there are some difference between the mutational drug resistance and transferable drug resistance. Generally, mutational drug resistance are single drug resistance. When compared to the transferable, it is multi-drug. These single drug resistance due to the mutational drug resistance will be of low degree of resistance, and it will not spread from one bacteria to the other bacteria because they may be defective and the virulence is also very low. So this type of resistance can be overcome by high drug dosage and giving combination of drugs. While comparing with the transferable drug resistance, generally they are multiple drug resistance and their virulence is not at all decreased. And since they are not defective, it can easily spread to one organism to the other organisms. There will be high degree of resistance. It cannot be overcome by high doses or even your combination of drugs cannot help here. So with this basics, we'll move on to the molecular genetics. So molecular genetics, the application of genetics in various other fields, which includes genetic engineering, gene therapy, DNA probes, use of blotting techniques, and PCR. So what is genetic engineering? It is nothing but a deliberate modification of an organism's genetic information by directly changing its nucleic acid genome is called genetic engineering. So a series of procedures used to recombine DNA is known as recombinant DNA technology. So these are the basic steps in genetic engineering. First, we'll isolate the gene of interest, insert that particular gene in a vector. Vector may be a plasmid or bacteriophage, and then insert the vector into a host. It may be E. coli or yeast cell. So produce as many copies of the host as possible. This is known as gene cloning, and separate and purify the product of interest. So that is what done here. First, you have to isolate the gene of interest, introduce into the vector from a bacteria, and introduce the recombinant DNA plasmid into the fresh bacterium and allow it to replicate. That is gene cloning, right? So after that, this particular organism will produce the product of interest. So where and all will you apply this genetic engineering? It is very much helpful in the medical field, food industry, industrial production, and environmental production. So as we know, in medical field, it is helpful to manufacture a lot of hormones, vaccines, and to correct endogenous genetic defects. It is known as gene therapy. So human proteins like insulin, growth hormone, parathyroid hormones, and a lot of products are produced by this technique. And certain vaccines like hepatitis B, rabies, Bordetella, influenza, most of the vaccines are produced by genetic engineering. And even vaccines for malaria and HIV are on trial on this method. So as we discussed, in gene therapy, we'll artificially replace the disease-causing gene with a normal allele. A normal allele can be carried by a virus vector to the target tissues. So it may be an ex vivo gene therapy, in vivo gene therapy, or even gene line therapy. So this gene therapy is very much useful in case of cystic fibrosis, rheumatoid arthritis, and vascular diseases in many of the cancers. So this genetic engineering is much useful in the food industry also. Almost 70 to 75% of the food in supermarket is generally genetically modified. So they are started producing hardier fruits, drug resistance, disease resistant, insect resistant crops with the help of genetic engineering. For example, they are producing golden rice, so that produce provitamin A. And these are the transgenic organisms generally produced with the help of genetic engineering. So these are few other examples for transgenic organisms. Regarding the industrial and environmental use, there are more efficient use of scarce minerals can be made, greater efficiency in mining can be done, and major concern in waste management. Biodegrade a number of waste products, sewage and petroleum products. So many bacteria can be engineered to eat the oil spills, thus reducing the environmental pollution. So genetic engineering is helping a lot to keep our environment clean and neat. Coming to DNA probes. DNA probes are nothing but radio-labeled or chromogenically labeled pieces of single-stranded DNA, which can be used for detection of homologous DNA by hybridization. So hybridization is nothing but a process of forming a double-stranded DNA molecule between a single-stranded DNA probe and a single-stranded target patient DNA. So this DNA is from target specimen, and this DNA probe is used for diagnostic purpose. So hybridization occurs between these two. So where and all it is applied? In clinical microbiology for diagnosing many microbial organisms in clinical specimen to detect microbes which are either difficult to grow or impossible to culture. So identification of culture isolates, we can use this method. Strain identification, we can use this method to identify toxins and virulence factors, and even to identify resistant markers. So next is a blotting technique. So immunoblotting is nothing but a highly sensitive and highly specific test to direct individual proteins in a complex mixture. So it's a combination of 3 different procedures. Initially, we have to separate the proteins and then do blotting technique. Separation is done on the gel and blotting is done in the nitrocellulose membrane. And after that, enzyme immunoassay or radioimmunoassay is done on the nitrocellulose membrane. So initially, Sir Edwin Southern discovered this technique. Hence, it is known as Southern blotting. The Southern blotting is done to diagnose DNAs. Later, they developed Northern blot to detect RNA and Western blot to detect proteins. And so there are many applications for this immunoblotting techniques to identify mutations, deletions, and gene rearrangements. They are used in prognosis of cancer and prenatal diagnosis of genetic diseases, leukemias, and diagnosis of HIV-1 and infectious diseases. Coming back to polymerase chain reaction. So we know that there is a process of amplifying a selected DNA fragment is known as PCR. So there are few steps in PCR. First step is denaturation of DNA. Here the double-stranded target DNA heated to form a single-stranded form. Annealing with primers. They are allowed to cool and anneal with 2 primers complementary to the flanking sequences. The third step is extension. The DNA polymerase and deoxyribonucleotide triphosphate are added and a new DNA is synthesized. So upon repeated amplification, we can detect this amplified products in real time. So it is widely applied in infectious diseases, diagnosing genetic diseases, neoplastic diseases, in forensic investigations, and an examination of phylogenetic relationships in evolution. So this technique is developed by Carey Mullis. So we discussed the same thing. First step is denaturation, annealing, and extension. And then the cycle repeats till the product is amplified to a desired quantity. So these are the different types of PCR. We do have a real-time PCR. Loop-mediated isothermal amplifications are done. Automated PCRs and automated real-time PCRs based on CBNAT. So this is an example for multiplex nested PCR system. Here we have 4 panels which are available for respiratory, gastrointestinal, meningitis, encephalitis, and blood culture identification panels. So this is a real-time PCR, which is used to amplify and simultaneously detect or quantify a target DNA molecule on the real-time basis. So we can skip the process of gel electrophoresis here. And this loop-mediated isothermal amplification, they are using this amplification method is carried out at a constant temperature of 60 to 65 throughout the PCR cycle. So as we discussed, this nucleic acid probes are of 2 different types, DNA probes and RNA probes. Thank you.